you on Planetterrian Daily, episode 93 for June 18th, 2026. Let's get into today's research and health developments. A spatial atlas now maps protein locations across 13,000 proteins in 3,000 human samples spanning 58 tissues and 25 cancers. Researchers generated high-resolution spatial maps from roughly 3,000 samples across 58 tissues and 25 cancers. The study quantified protein abundance and localization at high resolution across both healthy and diseased states. The resulting data set provides a reference for how protein organization differs between normal tissue architecture and tumor microenvironments. This resource can now be used to identify spatially restricted biomarkers or therapeutic targets that bulk proteomics approaches miss. Follow-up work will likely integrate these maps with single-cell transcriptomic and metabolomic layers. From tissue-scale protein organization, we turn to another genome-level insight into metabolic specialization. Sequencing of multiple sloth species identified expanded gene families that encode mitochondrial proteins involved in energy metabolism. The duplications may help explain the animal's characteristically low metabolic rate and slow movement. Researchers are now examining whether similar expansions occurred in other low-energy mammals. The finding links genome architecture directly to physiological specialization. Another crop genome now offers similar precision for breeding decisions. The new assembly resolves all chromosomes and captures both cultivated and wild peanut subgenomes in a single reference. High-quality annotation now allows precise mapping of genes controlling oil composition, allergenicity, and drought response. Breeders can use the sequence to accelerate marker-assisted selection for improved varieties. The work also clarifies the polyploidization event that gave rise to modern peanut. While genomes reveal what is possible, new tools are now automating how we measure individual particles. Researchers at the University of Gothenburg and Chalmers University trained an AI system to operate optical tweezers that capture, measure, and sort hundreds of microscopic particles per hour. The platform performs continuous sample loading, trapping, and data collection in a closed loop. By removing manual intervention, the method increases throughput for single-particle biophysical assays. The approach is now being applied to study protein complexes and extracellular vesicles. From lab automation, we move to organisms that endure extreme natural conditions without any assistance. South African-led sampling showed that sea ice microbial communities remain metabolically active through polar winter by synthesizing and catabolizing compatible solutes that stabilize proteins and membranes under extreme cold and salinity. These compounds allow the microbes to persist in a dormant yet viable state until spring light returns. The findings refine models of how polar microbial loops contribute to carbon and nutrient cycling. Cold tolerance in microbes contrasts with heat vulnerability in certain bees. A continent-wide survey of Australian native bees found that species nesting in plant stems lack the thermal buffering provided by underground nests. Tropical stem nesters were particularly at risk even though they already inhabit hot climates. The study measured nest temperatures and bee activity thresholds across multiple biomes. Behavioral traits such as nest depth and material choice emerged as stronger predictors of survival than geographic origin alone. 8 common food additives have been associated with higher cardiovascular risk according to recent analysis. More than 112,000 participants were followed for up to 8 years in the study. Higher intake of specific preservatives correlated with elevated incidence of hypertension, myocardial infarction, and stroke. Adjustments for diet quality and lifestyle factors did not remove the associations. These preservatives are widely used in processed foods to extend shelf life. The authors call for updated exposure limits based on these long-term observational data. From wild bees, we shift to everyday urban environments and the life they support below ground. Most people assume city environments are relatively sterile compared with forests or farms. In reality, the amount of visible greenery along streets strongly shapes the diversity of bacteria living in nearby soils and on surfaces. Right now, as you walk through an urban neighborhood, the density of trees and shrubs you see is quietly influencing which microbes can persist in that patch of ground. A recent study in Taipei found that neighborhoods scoring higher on a simple Green View Index hosted measurably richer bacterial communities even after accounting for parks and land use. Each incremental increase in visible greenery tracked with higher microbial richness, a pattern that held across an entire metropolitan area. The practical takeaway is that small-scale planting decisions, such as street trees, green walls, and pocket parks, can measurably affect the microbial environment people encounter daily. From space-exposed plants, we turn to cognitive variation already present in our closest relatives. Hundreds of wine grape seeds from Texas A&M will orbit for approximately 6 months. They will receive continuous cosmic radiation exposure before return to Earth. After landing, the seeds will be planted and grown to assess heritable changes in growth, fruit chemistry, and stress tolerance. The project aims to produce the first wine from space-exposed grape lineages. Researchers expect the radiation dose to induce novel genetic variation not easily generated by terrestrial breeding. From ape cognition, we move to the quiet mathematical lessons children encounter at home. Comparative testing across multiple ape species revealed consistent within-species variation in problem-solving approaches and social learning strategies. Some individuals relied more on individual exploration, while others copied conspecifics. The pattern mirrors human cognitive diversity and suggests these differences predate the human-chimpanzee split. Larger sample sizes across field sites are now being collected to test heritability. From household learning, we close with a national effort to account for ocean health. Observations in family kitchens documented frequent, spontaneous mathematical reasoning during cooking, measuring, and sorting tasks. Children encountered concepts of quantity, proportion, and pattern without explicit instruction. The study recorded these interactions across multiple households and age groups. Parents were often unaware they were providing foundational mathematical experiences. A multidisciplinary team produced an ocean accounting white paper that integrates ecological, economic, and social indicators for marine management. The framework will feed into the 2026 to 2036 National Marine Science Strategy. Pilot accounts are being tested in several coastal regions to quantify trade-offs between conservation and resource use. The approach aims to make ocean health data usable for policy decisions at multiple scales. The complete Spatial Proteomics Atlas spliced tissue and cell-level maps of more than 13,000 proteins across healthy and cancerous samples. It reveals how protein localization changes in tumors compared with matched normal tissue, offering a new layer of resolution beyond bulk measurements. Cancer biologists can now query whether a candidate target is confined to specific tumor regions or broadly distributed. The next step is integration with ongoing single-cell and spatial transcriptomic datasets to build multi-omic atlases that guide precision dosing and biopsy interpretation. The Spatial Proteomics Atlas and recent genomic studies illustrate how spatial and sequence data convert previously invisible biology into measurable reference maps. Next time, we'll be watching for updates on how the ocean accounting framework performs in its first pilot regions. That's Planetterrian Daily for today. If you enjoyed this, a rating or review on Apple Podcasts or Spotify really helps new listeners find the show. I'm Patrick in Vancouver. Thanks for listening, and I'll see you tomorrow. And before you go, this show is part of the NARA Network, a family of daily podcasts covering tech, science, markets, and more. If you enjoyed today's episode, give Modern Investing Techniques a listen. Modern strategies for Canadian and U.S. investors. You can explore the whole lineup at nernetwork.com. This episode used AI voice synthesis of my voice. Editorial selection and analysis are my own.